So we're going to take a look at the functional anatomy of prokaryotic and eukaryotic cells. To start out, guys, we're going to start doing a comparison where we're going to look at different structures and how they relate to prokaryotic cells and eukaryotic cells. Now, when we're focusing here on the prokaryotes, we're going to be focusing in on the domain bacteria. When we look at bacteria, their DNA is not found in a membrane-bound structure. It's going to be found in a general area called the nucleoid. We also see that their DNA is going to be in a single circular loop. So they only have one chromosome and it's going to be in a circular loop. Now in the picture it may not appear to be one single loop, but it's going to be wound up in that area of the nucleoid. When we look at the DNA in eukaryotes, it will be found in a centralized structure called the nucleus, and it will have a membrane around it. In the case for the eukaryotic cells, we do see that they have more than one chromosome. That's where the S comes in. They have chromosomes. And it is associated with these special proteins called histone proteins. Okay, so where the chromosomes actually wrapped around these special protein structures. With organelles, prokaryotic cells do not have any membrane-bound organelles. They're relatively simple internally in the fact that they don't have a lot of these structures. They do, however, have ribosomes present. And if you recall, ribosomes are important because they do protein synthesis. In the case of prokaryotic cells, these ribosomes are going to have a 70S structure, and we will talk about what that means a little bit later. When you go to eukaryotic cells, they are going to have various organelles. Now, organelles mean little organs, which means they have a lot of structures that are going to do different jobs. These are membrane bound. They also will have ribosomes, but their ribosome structure is a little different and it has an 80S structure. When it comes to cell walls, prokaryotic cells do contain a cell wall, and normally these prokaryotic or bacteria cells are going to have peptidoglycan found in their cell wall structure. Now, peptidoglycan is a very complex structure compared to the cell walls that we see in eukaryotic cells. Now, not all eukaryotic cells have a cell wall. Animal cells do not contain a cell wall. Plant cells do, and they have the structure of having it as cellulose, and fungi have a structure of chitin, and you can see that in the picture here. You'll notice that this chitin or chitin structure is not as complex as the peptidoglycan that you see for the bacteria. So a little concept here is that all cells do have a plasma membrane. Okay, the plasma membrane is the barrier of the inside of the cell versus the outside of the cell, but not all cells contain a cell wall. All right, so now we're going to look at cell division. In prokaryotic cells, the cell is going to go from one cell to two cells, and this process is going to be known as binary fission. Now, with binary fission, it is asexual reproduction because it does only start with one parent. Now, the two cells that come out of this particular procedure or process are going to be exactly the same as the parent cell. Now, eukaryotic cells also do a form of asexual reproduction where they go from one cell to two, but theirs is through mitosis. Mitosis actually means division of a nucleus. And so since the eukaryotic cells have a nucleus and it needs to divide, that is why it's called mitosis for them and binary fission for prokaryotic cells, because prokaryotic cells do not contain a nucleus. So again, asexual reproduction produces daughter cells, and those daughter cells are going to be identical genetically to the parent cell that we start with. Mitosis does involve the division of the nucleus, and this is why it happens in eukaryotic cells, whereas prokaryotic cells are going to use binary fission since no nucleus is present. Now, for prokaryotic cells in cell organization, they tend to be less complex and smaller in size because of this. And organisms that are in this group are going to be unicellular. When we look at eukaryotic cells, they are more complex, and this is due to the fact that they have all those organelles that are present. Most are actually multicellular. Now, of course, there is the exception in the kingdom Protista. But the cells of eukaryotes are normally about 10 times larger in size of the cells of prokaryotes. All right, so we're going to mostly focus on prokaryotes in this particular presentation. We'll focus more on the eukaryotic cells a little bit later in another presentation. So with prokaryotes, this is the domain Archaea, which none of them cause disease, so you're not going to hear about them much throughout the course, and bacteria. Now, it is important that we ID the type of bacteria by cell shape and arrangement. So when we're looking at shapes, there's going to be a series of shapes that we're going to be looking at. And there's three kind of main groups for the most part. We have the round shaped cells. These round shaped cells are known as coccus cells. And I have a little O highlighted there so that you see that it's round. Okay, so these are the round shaped cells. And when we talk about the word coccus, here it kind of means berries. We then have a type of rod shape. This is known as the bacillus. And so I have the two L's highlighted to show you they're rod shaped. All right, so we have that bacillus group. Now you'll notice they're not all exactly the same shape rod wise. Some of them even look like little drumsticks or whatever, but they do have that rod shape. The last group are going to be the helical, the ones that look like a spiral. And so these are going to be spirillum. Now spirillum are going to be more rigid in their structure, whereas spirochetes are going to be more flexible. Also in this group, there is what we call the vibrio, and vibrio have a comma shape to them. So this is what we're looking at for the shapes. Now, what about arrangement? Now, if you'll look in this particular diagram, you're going to notice that these are all cocci cells. So they're going to be all round cells. So let's look at some arrangements that these cells can undergo. We see that they can have the diplo. This means they're in pairs where two of them are together. So we would call this diplococci because this tells us there's two and they are those round cells. We also have the strepto. Strepto means it's in a chain. And if you look here, we have a chain of those circular cells. So this is streptococci. Now, sometimes they'll come in these kind of packets or groups, and we see that a tetrad is going to be in fours, and a sarcony is going to be in cubes or octets. The last one is staphylo, and staphylo means grape-like clusters. And so in this case, you have a cluster of these round cells, so it's called staphylococci. Now, these may kind of sound familiar. If you've heard of somebody who had a streptococcus infection, this is what it looks like underneath the microscope. If they have a staphylococci infection, that's what we're looking out there down at the bottom. So this gives us an idea of what cells are present, but also the arrangement. Now, a quick thing to note though, guys, that organisms that are grown on artificial media, like in the plates in the lab, they may not typically exhibit this cellular arrangement. They may not get into their strepta or their staphylo as well as if you were to take the sample straight out of it from a living organism. So you may not see streptococcus actually in chains when it's grown on artificial media. The same thing with staphylococcus, you may not see them in the clusters as much. So you'll notice here that you'll see that this is when gram staining from an agar plate, you'll notice that they look very similar, okay, in that structure. Versus if you look at the other two pictures that were out of the living tissue sample, you can see that there are some differences. You see the chains versus the clusters. All right, so now using the information that we've already gone through, describe the following by gram reaction. So is it gram positive or gram negative? The morphology, so that's telling you the shape of the cells, and then the arrangement of the cells. Okay, so you've got to be able to do this, okay, when you're going through this course. So in this first example, we see that these are gram positive. We know they're gram positive because they are purple. Okay, so we're looking at the smaller cells here because bacteria is smaller. They're prokaryotic cells. And so these are going to be gram positive. If you'll notice, they're in twos. So that's the diplo, and they are circular. So that's the cocci. So this is gram positive diplococci. The next one, because it has a pinkish red color, this one's going to be the gram negative. Again, they are in pairs and they have kind of this roundish structure. So in this case, we have gram negative diplococci. The last one is gram positive. Again, it is purple in color, and it's going to be the cocci in chains. And so since the cocci is in chains, we'd say that it is streptococcus. So when we look at this, guys, we need a definitive idea of the bacteria cannot be made solely on the gram reaction in the morphology. Okay, we can't go through the process and be like, oh, that is what that particular one is. We've got to have to do further tests to find out. The reason being is when we look at this gram-positive diplococci, is it Streptococcus pneumoniae? Well, wait a minute. The strepto tells you it should be in chains. Okay, but is that what we're looking at? The gram-negative diplococci, is it Neisseria gonorrhoeae? Don't know. We have to do some further tests. And of course, then the last one, it could actually be Enterococcus, not Streptococcus, when we're talking about it as a species. All right, so it's important for us to do more tests in order to figure out what potential bacteria we're looking at. All right, so when we move on to the bacillus group in the sense of the rod-shaped cells, their arrangements are going to be a little bit different. Okay, so we see that these terms are typically not used to describe cellular arrangements for bacilli. We don't normally use diplobacilli and streptobacilli when we're describing things like we did with the coccus group. All right, but they are present. They can be in twos or in chains. We also see that sometimes if they have kind of a shape that's similar to cocci but also bacillus, we give it that name Cocciobacillus, okay, because it's kind of a mix of the two. Now guys, one thing that's important here is the term bacillus is used to describe cellular morphology, but it is also going to be used to describe some species, and these are called the bacillus species. If you'll notice though, there is a difference. When we're talking about a particular species, what do we have to do with that name? We have to capitalize it. Okay, so that genus Bacillus is capitalized, so this tells you we're talking about something specific, not just the general cellular morphology. All right, so describe the following by Gram reaction, morphology, and arrangement. Okay, so in the first one, we have that purple color, so these are going to be gram-positive bacilli. They kind of have a boxcar shape, okay, but these are gram-positive bacilli. The next one is also purple, and these are gram-positive bacilli, but you'll notice these are in chains. We're not going to call that streptobacilli, but they are in chains. Now, if you'll look at these two that we have here, you'll see that these are considered monomorphic. Now, monomorphic means you have a single shape that's present within the culture. On the other hand, over here, we have gram-positive, but we have what we call a plelomorphic bacillus. This is described as looking like kind of Chinese letters or clubs like in palisades, which are like evenly distributed cells. A lot of times these are also called diphtheroids. An example of these are going to be Corynebacterium. All right, so now we're going to actually take a look at structures that are going to be present in some of the bacteria that we will be studying. Now, these structures we're going to start with are going to be on the external part of the cell wall. The first one is what we call a glycocalyx. Now, glycocalyx is can be a capsule. The capsule helps with virulence, and a capsule is firmly attached to the cell wall. On the other hand, we can have a slime layer also out here. It's a type of glycocalyx, and it's only going to be loosely attached to the cell wall. So if you'll take a look here at this picture, we have the inner plasma membrane in the yellow. We have the middle cell wall area that's in the green. And then we have the outer capsule, which is the orange. In this case, it's firmly attached to the cell wall. Now, this is a gelatinous kind of sugar coat around the bacterium cell, and it protects the cell from being engulfed. This is going to help protect it like from your immune system. Your immune system's maybe going to have a harder time holding on to it because of this gelatinous type of structure that's around the cell. An increased virulence factor means that it has an increased chance of making you sick. It has an increased chance of being pathogenic. Now, sometimes there's what we call a K antigen. The K antigen is a capsular antigen, and this actually induces an immune response in some cases. What types of staining allow us to be able to see capsules? We talked about this in the previous presentation. So what type of staining is this? This type of staining is the negative staining technique. So guys, here's some examples of some bacterium that are going to have that gelatinous capsule to it. And you can actually see it when they're growing on the plate. You can see that it has more of a jelly-like substance to it or texture to it. Klebsiella pneumoniae on the McConkey's agar over here. And we have also Streptococcus pneumoniae on the sheep blood agar, okay, the 5% sheep blood agar. So it shows you how they're growing. So the colonies kind of have this mucus-like structure to them, so mucoid looking structure. Some virulent strands of Bacillus anthracus is also going to produce a capsule. The next one we want to look at are the flagella. The flagella are going to help with motility, movement. Now these are associated with an H antigen, and so when you see examples like with this E. coli where it says E. coli survar O157H7, the H is referring to the type of flagella antigen that's present. Okay, so it could be different based on like mutations that can occur even within different subgroups of a bacteria like E. coli. So the first flagella we're looking at is going to be the Prokaryotic type of flagella, and that is going to be where there are many flagella and they're all over the organism. We then have the monotrichose and what also is called polar. So when it's a mono, it tells you that it only has one, and polar tells you it's on one end. The lophotrichose are going to also be many, but they're also going to be contained in one area, and so that's again where the polar is located. So it's going to be on one end. The amphotrichosis are also going to be polar bacterium, and you're going to see the flagella on both sides. Okay, so on both poles, the flagella will be present. Now, a lot of times, guys, with this flagella, it helps it move, and it does it by rotating, and the flagella can either rotate clockwise or counterclockwise. We also see that there could be axial filaments for motility. These are going to actually run and wrap around the whole body of the bacteria. This is actually what you see in spirochetes, okay, in that spiral type shape, and this is what allows it to move more of like in a corkscrew type of movement. An example of this is Treponema pallidum, which causes syphilis. We also call a lot of times these axial filaments endoflagella because the fibrils wrap around the cell and lays within the outer sheath of the cell. Another structure that we want to look at are what we call Fembrae. Fembrae are going to have two kind of functions. For one, they're going to help with adhesion and holding on to the bacteria being able to hold on to something, as well as a sex pilus. And guys, the sex pilus part of a Fembrae is going to help transfer genetic information from one bacteria to the next. So remember, they're going to reproduce by asexual reproduction, which means they all would be the same, but mutations do happen, or they pick up pieces of DNA that help them, and then they can pass them on to bacteria that are nearby. Now, Fembrae, guys, are going to be bristle-like fibers that extend out from the cell. A cell may have 200 to 400 of these, and it allows them to attach to surfaces. Now, when we look at this, it acts kind of like a Velcro in that sense, so it allows it to attach. When we look at the sex pilus, it has more of a tubular structure. It will be longer than your normal Fembrae, okay, and we do see that they'll have none or less than 10 of these. And the whole point of this is, again, for genetic information transfer, and that is called bacterial conjugation. If Fembrae is present, it's associated more often with gram-negative organisms. So some examples would be things like E. coli, Neisseria gonorrhoeae. The next one we want to look at is the cell wall. The cell wall protects the plasma membrane and also provides and gives shape to the cell. Now, this cell wall can be gram-positive, which we talked about previously. If it's gram-positive, it's going to stain in that purple color due to crystal violet, and it's going to have a thicker layer of peptidoglycan, which you can see here in the picture. Gram-negative, on the other hand, is going to have a thinner layer of peptidoglycan. It's also going to contain an outer layer of what we call lipopolysaccharides. Now, these lipopolysaccharides, also referred to as LPS, are going to be important because a lot of times they are going to produce endotoxins. Now, an endotoxin that could be released is called lipid A, and it gets released when the cell dies. So what happens here is that the cell is the kind of making you sick, and it triggers your immune system, and then your immune system attacks it and destroys it. But the bacteria is like, oh, wait a minute, I got something else for you. You destroyed me, but I'm going to get the last laugh because I'm going to release this endotoxin, which is going to cause you some more problems. Okay, and so again, it's a virulence factor, but it's going to be a toxin found inside of the cell wall, and when the cell wall ruptures, the toxin comes out. Another one is an O polysaccharide. The O polysaccharide is kind of antigen, and again, it can help us be able to distinguish different subtypes of a bacteria, like with E. coli. So that O157 is referring to the type of polysaccharide that is present here. Now remember, gram-negative bacteria are going to stain more of the pinkish color because they are not going to hold on to the crystal violet due to the decolorization in the gram stain, and they are going to take in the secondary stain, which is the safranin. Another thing that is important with these gram-negative bacteria is because they have the peptidoglycan with this lipopolysaccharide structure, the lipid polysaccharide structure actually helps act as a better barrier than just the peptidoglycan, and it protects against detergents, heavy metals, and bile salts. And so it's going to cause it to be harder to potentially treat because it's going to be harder to access. Even while some antibiotics have a harder time working on gram-negative bacteria. When we go back over here to this endotoxin that could be released, remember I told you it's kind of like the bacteria gets the last laugh. It's like, hey, you killed me, but I'm going to still cause problems. Some of those problems that like lipid A can cause are going to be things like fever. They could send the person into shock, and blood clots. Now, another type of bacteria that we need to look at are called mycoplasma. Mycoplasma is the causative agent of walking pneumonia, and they are a type of bacteria that's kind of an exception to the rule. Remember earlier we talked about prokaryotes and bacteria pretty much all have a cell wall? Well, mycoplasms do not. They lack a cell wall. They're the smallest living microbe that's out there, and they have a high lipid content. And this lipid content contains a lot of sterols in their plasma membrane. Mycobacterium also has that high lipid content, and this is going to be what we call that myclonic acid. Now remember, with the myclonic acid, we have to stain it with a different type of stain. Gram stain doesn't work, and we have to use that acid fast. Now, archaea, remember, is not talked about much here, and one reason why archaea bacteria were moved to different groups is because their cell wall structures were so different. Another note is that some antibiotics, they actually interfere with cell wall synthesis. They interfere with the bacteria making the cell wall that it uses for protection. Penicillin affects those cross bridges that are found in the gram-positive bacteria of that peptidoglycan cell wall. But penicillin does not work near as well on gram-negative bacteria. So bacteria cells with atypical cell walls do not gram stain well. Okay, so if their cell walls are different, they don't gram stain real well. Different antibiotics have to be used on gram-positive versus gram-negatives because of the structural differences that are present. And damage of the cell wall can lead to osmotic rupture of the plasma membrane. Guys, if we can poke holes in their cell wall, it makes that bacteria more vulnerable, and it makes it where it's more vulnerable to losing water or gaining water, and it can cause the cell to rupture. All right, so let's do a quick comparison here of gram-positive versus gram-negative. When we look at the gram reaction, we see that it's going to retain the crystal violet with gram-positive, and it's going to take the counter stain of safranin if it's gram-negative. The peptidoglycan layer is thick in gram-positive, it's thin in gram-negative. When we talk about the periplastic space or outer lipopolysaccharide, the release of endotoxins, we see that that is absent in gram-positive bacteria. But in gram-negative, these are present, and they release that lipid A endotoxin. Now, sensitivity to what we call lysozyme, which is a natural type of antibiotic that is made mostly like in your tears and some of your bodily fluids, as well as penicillin. Gram-positive responds very well to it. It actually can be destroyed pretty easily and weakened by lysozyme and penicillin. Gram-negative, however, its response or sensitivity is low. Now, resistance to drying out. Gram-positive has a high resistance to drying out, but gram-negative does not. Now, this means that if they are able to not dry out as quickly, they can actually be in the environment for longer and be on surfaces longer. So gram-positive bacteria can survive on surfaces longer because they're not going to dry out as quickly as you see the gram-negatives. All right, so this brings us then to plasma membranes. Now, the plasma membrane is also known as the cell membrane or the cytoplasmic membrane. And we see the structure of the plasma membrane is kind of like a fluid mosaic model. So guys, when we talk about a fluid mosaic model, it is a phospholipid bilayer, meaning there's two layers of those phospholipids where they have the heads and the tails. It does create this fluid type of structure. It's kind of like a lipid sea, a fat sea here. The mosaic part is telling you that there's a bunch of stuff that is embedded within this membrane. And in this case, it's going to be mostly proteins. When we talk about bacteria plasma membranes, they actually do not contain certain structures like animal plasma membranes contain. They don't contain the glycoproteins, the glycolipids, or the sterols, okay, except for the mycoplasm that we talked about earlier. They do retain, however, the integral proteins, the peripheral proteins, and the surface proteins. So they contain still all those protein structures, they just don't have the others. Now remember, when we have this phospholipid bilayer, the head of the phospholipid is hydrophilic, liking water. The tails, however, are hydrophobic. So what are the functions of this plasma membrane? The plasma membrane acts as a barrier between internal and external environments. It is subject to rupture if it's not protected. And so this is why bacteria really wants that cell wall. The cell wall helps protect it from rupturing. The other part of function of the plasma membrane is for transport. It's going to allow the movement of substances into and out of the cell. Now, it's not just going to let anything move in and out. Something that's really important about this is it is selectively permeable. It's going to pick and choose what gets to move in and out of the cell. A lot of times I compare it to like a bouncer of a club, like one of those high-end clubs, and you have the big line of people. They're going to pick and choose who gets to come into the club. Okay, that's kind of how the cell membrane is going to work, because it wants to bring in the things that are beneficial to the cell, and it wants to get the things out that it needs to get out of there, whether it's waste products or things it's made that need to be released. Okay, so it's going to be selective on what gets to move in and out. When we're talking about bacteria cells, the plasma membrane is where aerobic cellular respiration is going to occur. Cellular respiration, guys, is how the cells create ATP. And for us, that happens in the powerhouse of our cell, which is the mitochondria. Remember, bacteria, though, don't have organelles. They don't have mitochondria, so they are going to have special proteins embedded in their membrane, which are going to allow them to do cellular respiration. All right, let's talk a little bit about this transport of moving in and out of the cell. Now, there is what we call active transport. Active transport is going to be where ATP or energy is required to move substances. And the reason why ATP is required is because we're going to try to move substances against their concentration gradient. Guys, most substances like space. Just like us, we like our personal space. Okay, so do molecules. So if they're tightly packed together, those molecules want to spread out. Okay, and the idea of spreading out is they're going to go from high concentration to low concentration. Well, in active transport, we are going to push them back to the high concentration. We're going to push them where they don't want to go, which means that it requires energy. Okay, it's kind of like if you've ever seen a parent or a person deal with a toddler who does not want to go somewhere. That parent has to utilize a lot of energy a lot of times to move that child, because the child acts as dead weight. Okay, and so that's kind of what's happening here, is the cell's having to use energy and push it to the area it does not want to go. Passive transport, on the other hand, is no ATP required whatsoever. This is just letting the molecules move from high to low, okay, where they're spreading out. This is like getting in the lazy river at a water park. You get in, you don't have to do anything. The current does all the work for you. Okay, and that's what it's looking at for passive transport. So guys, here are a lot of examples of passive transport. The first one is what we would call simple diffusion. Molecules are just moving across that plasma membrane from high concentration to low concentration. Okay, they don't need any kind of help. They can just do it through the membrane. However, some molecules are either too big or they have a charge that makes it harder for them to move across the membrane, and they need some help. In that case, they're going to use those transport proteins that are embedded in the membrane. That's called facilitated diffusion. They're going to get some help, but again, still no ATP is required because it's still moving from high to low. The last one over here is the movement of water, and the movement of water from high to low, the diffusion of water, is called osmosis. Now, why do we need to talk about this, and specifically that movement of water? Well, osmosis is movement of water from high to low, okay, so it has a concentration gradient against that plasma membrane, that selectively permeable membrane. Now, when we put cells in different solutions, we call that tonicity. The tonicity of a solution describes the solute's concentration, and it is relative. Okay, so in this particular case, we have a hypertonic solution. Okay, the blue solution has tons of little green guys in there. Okay, those are the solutes that are placed in there. Okay, so there's a lot of them. So this is hypertonic, more solutes, less water. The cell then, however, is hypotonic because it has less solutes. On the other hand, this picture is showing you a hypotonic solution because there's only one little solute in it versus the cell, which is hypertonic. If it's the same, there's equal amounts on both sides, we call this isotonic. It's the same. Now again, why is this important? Well guys, water is going to move from a high concentration to a low concentration. So in the case, guys, water is always going to flow from the po side to the per side. It's going to flow from the hypo to the hyper. So in this case, water is going to go out in the first picture. This is going to cause the cell to shrivel up and shrink. That cell then could die and not continue to do its job. In the second picture, the water is going to go into the cell. This could cause the cell, if it doesn't have that protection of the cell wall, it could cause the cell to expand and eventually burst. Okay. Now in the third one, with it being isotonic, the water is just going to move back and forth at an equal rate. Okay. No change to the cell is going to occur. Now, cells placed in isotonic solutions have no net movement of water across the membrane. So water will move, but if 5 move in, 5 move out. 10 move in, 10 move out. There's no net gain or loss. But cells placed in a hypotonic solution will swell, and osmotic lysis could occur if that cell wall is damaged in bacteria cells. This is why taking certain antibiotics or lysozyme activity is important, because if it pokes holes in the cell wall, then your body can use this structure in order to cause the cell to burst. On the other hand, if a cell is placed in a hypertonic solution, it'll shrink up, and this is called plasmolysis. You can see that the plasma membrane is pulling away from that cell wall structure. Again, the cell can be rendered useless here and it could die. So an application, well, this whole idea of water and the movement of water in different solutions could control microbial growth. In order for bacteria to grow properly, they need perfect osmotic conditions. So this is why when we do cultures and we want to grow it in the lab, we need to give those good conditions on the media. Bacterial cells in a hypotonic and hypertonic solution are subject to becoming metabolically inactive. Therefore, they can't be utilized anymore. They can't continue to do their job because their metabolism has stopped. All right, so some other structures we want to take a look at. This is moving into the cell. We have the cytoplasm. The cytoplasm is composed mostly of water with a variety of inorganic and organic substances. It's an aqueous solution, and it's going to end up bathing the whole DNA area, that nucleoid. And this is also where the ribosomes are floating in the cell. The nucleoid, recall, is the region or the location of the single loop of DNA. Now remember, since this is still DNA, deoxyribonucleic acid, it is still double-stranded. Okay? It has the two sides, the twisted type ladder structure to it. Sometimes, though, these bacteria will have small pieces of DNA that are not actually connected to that single chromosome in the nucleoid. These are an accessory ring of DNA, and these are called plasmids. These are not crucial for the metabolism of that particular bacteria, but they're extra genes, and these extra genes could be donated through that sex pilus we talked about from one cell to the other. These can be responsible for antibiotic resistance, guys. This is how they share that. Then when one's like, hey, I don't get destroyed by that antibiotic. Do you want protection? And the other cell's like, yeah. It gives a piece of that DNA and it helps spread that. This is also how some of them start producing toxins and other virulent factors that cause them to be a little more dangerous. We also use plasmids in biotechnology when we want to do gene manipulation and transfer. Okay, so we are taking what they automatically do and we are manipulating it and using it in a different manner. All right, let's talk a little bit about these ribosomes. Ribosomes, remember, are responsible for protein synthesis, and they are made up of two subunits. They have a large unit and a small unit. Now, one thing that is really important is the fact that our ribosomes are different than the ribosomes found in bacteria. This allows for the use of certain types of antibiotics. We talked about some antibiotics mess with the cell wall, which is fine because in animal cells like us, we don't have a cell wall, so it's specific against the invader and not us. But what if we want to attack the ribosomes? We have ribosomes and so do they. Well, our ribosomes are a little different. Antibiotics can be used to target that 70S structure versus the 80S structure that our ribosomes have. So if you take a look at the prokaryotic ribosomes, they're a little bit smaller. They have a 30S small unit and a 50S large unit. When those combine, they make a 70S unit. And you're like, wait a minute, that doesn't add up. I'm not even sure why they don't add up that way, except for that when they come and combine together, their sedimentation rate does not equal what it did before. Now, sedimentation means when they're spinning it in a liquid, how quickly does it pull and fall to the bottom? Okay, like how heavy is it in that sense in that liquid? In eukaryotic cells, our ribosomes are larger. Our small unit is 50S, our large unit is 60S, and when those combine, it makes it 80S. So these are unaffected by the antibiotics. So again, when we talked about medication and chemotherapy, it's important for medication to be specific in order to be effective. So these antibiotics are going to trigger or going to attack the 70S unit, but they don't attack ours. They don't attack those 80S units. So our cells can still make proteins, but the bacteria cells cannot. Another structure that bacteria can end up having are called endospores. Now, endospores are metabolically inactive structures. This is where the bacteria becomes dormant, like it's sleeping. Okay, these are dehydrated cells and they have a thickened layer of peptidoglycan to protect them against like hostile or unfavorable conditions. Now, these cells can't reproduce because their metabolism is so low, but it does allow them to survive while those harsh conditions are present. These are very hardy, guys, and they can actually survive in the environment and on surfaces for many years, okay, because they are going to have this dormant structure. However, when the environment is better for them, it can trigger them to become where they start germinating, and when they germinate, they return to the vegetative state and they can do metabolism and they can reproduce. Okay, so this causes them to be an issue because they could sit dormant for several years and then potentially make you sick. All right, so that's how endospores are an issue, and they are an increased virulence factor. In order to help make sure infections don't occur with organisms that have endospores, we must kill them during a sterilization process. We got to make sure that the sterilization can actually penetrate and get through that endospore structure. So some examples with endospores, we have here gram-positive endospore producing bacilli. This is like Clostridium. Clostridium causes botulism, tetanus, and gas gangrene. Lots of things you do not want. Okay, botulism is going to actually release a toxin that causes your muscles to relax. Okay, we actually use this for Botox. Okay, they use the toxin from Clostridium to do Botox. The issue is if you have the bacteria, it keeps making the toxin, and it's not so much of an issue until it gets to your diaphragm and then you can't breathe because it can't contract. On the other hand, tetanus releases a toxin that causes your muscles to contract violently. It causes you to have all kinds of major cramps, and those can actually be so severe they can break bones. And gas gangrene, guys, it's terrible. It eats away at the tissue, producing a toxic gas that smells terrible, but also pulls away the layers of tissue, causing more damage. We also see some bacillus species will have these endospores. Some cause food poisoning. And then anthrax is also a part of this. And you've probably heard a little bit about anthrax, or at least in the past, where it was mailed to like the Pentagon and things like that, and it caused severe illnesses. That's why in some places they treat the mail to try to eradicate any endospores that could be transported. So implications, guys, the big thing here is proper food preservation, like canning needs to be done properly, because if you don't and these endospores are in there, they can survive for a long time. And also the importance of vaccines and public awareness. It's important to make people aware of what's going on, and that whole idea of using proper techniques with preservation of food and vaccine. Now, eukaryotes, guys, we are going to focus more of that on another presentation that we'll do over chapter 12. But remember that we're going to be looking at protista and focusing mostly on the protozoa with a few algae. Fungi, we're going to look at yeasts and molds. And with animalia, we're going to look at the helminths, the parasitic worms. Remember that some of these will also have flagella, cilia, and cell walls, similar to what we talked about in this presentation. But all of them do contain a membrane-bound nucleus and organelles because they are in the eukaryotic group. They're eukaryotes.